Good morning, everybody. Thanks. I want to thank the organizer for inviting me here. And I would like to talk about our products or ideas or research. I am from IMIGO. IMIGO is about sensors, as you can see. As you can see here down here, solutions beyond sensors. And I will talk a little about the ideas of transferring research, perhaps, from university to market. IMIGO business models, and IMIGO as such, we are about 40 employees, have a turnover of 4.5 million euros. We have industrial collaborations where basically industry hires us to solve problems. R&D projects, licensing, royalties, and spin-out. And I start with spin-out. No, I start with presenting what we do. We are kind of spread out organization. We are very unfocused. We have R&D in bio, in electromagnetics, photonics, in MEMS, packaging, and so on. And then we have a huge system, well, not huge, but large system design group, which converts these pieces or small or ideas or components into systems, because it's only the systems which you could use really in practice. And so these two groups merge and collaborate in order to create what we call technology platforms. Basically, it's the platforms which will sort of cheat the customer, because we will use the same platform for a lot of applications, and thereby, hopefully, or really, get some revenues out of our activity. So what we do is, in practice, is to detect bacteria. We are in clinical diagnostics, but also in processes and in industrial processes and in industrial solutions. And what I show now is an example of spin-out, which is based on our IP. It's a remote sensing of the speed and of the pressure exerted on a huge fabric on which cellulose is running. It's for a paper industry. Paper industry is important in Sweden and, for example, in Finland as well. And what we do here is simply include piezoelectric fabric threads into their fabric, and then collect the signal from the threads when the fabric passes through these pressure cylinders. And thereby we can determine the velocity and also the parallelity of the cylinders and decrease wear of the fabric. It costs about 10,000 euro per hour to exchange such a fabric, and it takes 2 days, and the fabric itself costs about 250,000 euro. Next thing I would like to talk about is IMIGO's inertial sensor systems. We are very strong in inertial systems. We started with components. We started basically with our IP on 3D accelerometers and on gyroscopes. And I believe that those who had, well, it's not so popular anymore, but those who have Volvo or Saab or a lot of other cars, actually Fiats as well, probably do not know, but the cushions and the curtains which are in the cars contain our components. 70% of them do. We started with this in 90s, and the picture here shows the development. It took several years before we had a system going on. And that's on the right-hand side, where we have designed an ASIC that would convert the component 3D accelerometer into a useful system. And then we connected all of these into another system which still produces still more work, namely motion tracking and inertial navigation. And I will skip several slides and go into something. And the example of the performance of the systems is the measurement of the rotation of the Earth. As you can see, it's pretty robust system and pretty sensitive. We use it in different applications, and some potpourri of the applications is shown here. For example, we have made specific packaging so that the system would fit into drilling bores. And so we could monitor how the drilling bore is moving when, for example, searching for oil. We have also this item installed into a testing doll for testing the collisions in the cars, and now working on the single person inertial navigation and motion tracking for firemen. As you see, we collaborate with the companies who use these, either on licensing basis or we just produce them and sell. As a next application, I would like to switch the field completely. This is into biotech processing. It can be used to detect, for example, harvested proteins during protein production. It takes about 10-15 minutes, and you will get protein concentration. And so when your yeast or bacteria start to deteriorate and actually consume the proteins, you can break the process up. Ordinarily, it takes about 4 hours to get the results, so nobody uses it, but this takes about 15 minutes. The idea behind is to use Brownian motion of magnetic nanoparticles. And we have also built systems which we sell and had sold, well, 3 or 4 systems. It's detached system and works completely well. And the examples I have shown here are the detection of DNA, the hybridization of DNA. On this slide, as you can see, we can detect it pretty sensitively. And also an application when you determine the antibodies of the Brucella abortus in milk. This is quite an important disease in cows, and we have collaboration with an Indian company which will use it in their products. Next example is actually a prototype or a demonstrator that allows one to diagnosticize prostate. It works on a very, very conventional basis. The only thing which is special about it is the fact that all the fluids, all the rinsing fluids and all the waste are contained within this small chip, which is rather cheap and is disposable. So there is no spill out, and that's quite important if there are bacterial diseases. Right now we collaborate with Norwegian company and also with a Swedish one, again Indian one, in order to adapt this system for TBC detection. 2 minutes. Okay. So I will go very fast. We work in wireless technology as well, but this is more of a developmental project where we have concentrated on a very, very cheap and low power consuming RFIDs, which are passive, so they are pretty cheap, but have some intelligence sensing. Finally, we work with gravimetric techniques where we simply measure mass or viscosity, and there we have used quite a lot magnetoelastic resonance and can wirelessly transfer various types of signals, for example, during paint hardening, and that's in real time, as well as the growth of biofilms. In this particular case, this was E. coli, and we functionalize surfaces so that we do phenotype-specific cell detection or biofilm growth. I skip this. Then we work, that's also more of an R&D project. We started to work on receptor-ligand interaction and forces, but there we use our own approach. We simply try to functionalize magnetic nanoparticles and then pull them away when they are adsorbed on surfaces. Thereby, we can decrease non-specific binding and also determine the strengths of the receptor-ligand interaction. Finally, we work quite a lot on something called molecular imprinted polymers. These are used, for example, to detect pharmaceutical waste in water, but also other compounds, chemical warfare agents, and so on. And what we do here is, again, to develop techniques for sensing, whereas the materials themselves are provided to us by our collaborators. And there I finish my presentation. This is just to show the performance of the inertial motion system, which I have shown. And as you see, we can write Amigo quite well. Thank you very much.
Thank you very much.